Thank you, Francesco. Thank you all for coming. So yeah, I'm going to talk about this dynamical effect of hierarchy of timescales in the brain. So first of all, what's a hierarchy of timescales? So this is an interesting framework. And so Rita and Danny Bassett presented keynote talks on similar topics, but not together. So Rita was talking about hierarchies and the importance of timing, and Danny Bassett was focusing mostly on the role of network topology. So if you put together this hierarchy, this timing, the importance of timing, and the role of networks, we get this. We can think about this hierarchy of timescales. So I'm going to argue that this is a general organizational principle of the brain, and this is very simple. So it's just a gradient between regions that have slow and fast timescales. So if we think about the connectome, we are going to see hub regions that are more stable, would have more slow dynamics, and peripheral regions that have more fast dynamics. So this is a hierarchy, or this is the gradient that gives rise to this hierarchy of timescales, and seems to be very general across species and modalities. And I'm going to go through these and try to convince you guys of that. So yeah, the brain has to cope with events that vary over several orders of magnitude. So the spike of a neuron's 1 millisecond is probably one limit, but the perception takes place at hundreds of milliseconds. And there are other events that vary over much longer periods of days and weeks. So for instance, mood evolves in this very slow time scale. So if you think of the brain as a dynamical system, it's very impressive that it has to respond to events that vary over so many orders of magnitude. And if you think so this is actually a very relevant or interesting and hard question, how a dynamical system can respond to events that vary over so many orders of magnitude. But then we can also think of the brain as a physical system. So it's a physical entity and has brain regions, and it's a network, and it's very important to take into account that the brain is heterogeneous, that there are different regions, and some regions are highly connected with other brain regions. Other regions are more peripheral, and they're not as connected. So these highly connected hub regions, they form a rich club. They are also connected to one another, and they form this core, this rich club core. And they are connected to feeders and peripheral regions. And I'm going to first address this with this perspective, or with a computational modeling perspective. Later on, I'm going to go to more empirical data with fMRI and TMS experiments. But I'm going to first start with this simulations computational modeling approach. So in this case, we have different regions in this connectome, and we can simulate the dynamics of these regions with neural mass models. We connect them according to the connectome, and we are going to look and analyze the emergent dynamics of this network. So if we simulate this, we're going to use this BTF model. It has two time scales. It's a fast rhythm of gamma band and a slow rhythm of alpha band of about 100 milliseconds. And this is the dynamics we get. This is an illustration of three nodes: a rich node in red, a peripheral node in blue, and feeder in red or in black. And these are the equations. This is a somewhat standard model. And it's important that in this network, we are going to assume that all the nodes are identical. That's the most simple approach. And the relevance of this is that if you assume everyone is identical, all the difference that we observe can be ascribed just to the connectivity patterns, to who you are connected to. That's the only difference that one observes in this network. So if you look into the dynamics of the whole brain, what we observe is something like this. So some regions, the regions synchronize at this fast rhythm, and they are out of phase in this low rhythm. So you can actually look into which regions have this peak together. So we can define functional clusters as these regions that are synchronized, that are having this peak together. And we can keep track of this dwell time of each region in these functional clusters. So if we do this, for example, we can see that the period of this functional cluster is about 100 milliseconds. This is this low rhythm. But one region can dwell in this same functional cluster for several seconds, so for 50 or 100 periods. So this is an example of a rich club region that's very stable. It can dwell in this functional cluster for multiple seconds. And in contrast, we see when we observe that peripheral regions, they tend to be much more flexible. They tend to change between functional clusters much more often. So there is this gradient, in which you have stable core regions and flexible peripheral regions. So this gives rise to this structure dynamics interplay, in which we can relate the features of the topology of the network with features of the dynamics, of the emerging dynamic of this network, with flexible regions being, or peripheral regions being flexible and core regions being more stable. So this is also consistent with our understanding of the brain. So this is based on neuroimaging experiments, for instance. We see that some sensory motor systems is more associated with our perception and our motor and our relationship between the brain and the external world. And in contrast, we have core regions that are more associated with autonomic functions and emotions and mood dynamics that have slower dynamics. So there is this relationship between features of the connectome, levels of the hierarchy of the connectome, and the interactions of the brain with the external world and the internal body. So this hierarchy of time scale is also seen and described in many other systems. So this is an example in single neural electrophysiology experiments in macaque. So in which one can look into the autocorrelation of these signals and measure and estimate this autocorrelation or time. So this intrinsic time scale, so the decay of this autocorrelation function. And there is a big diversity, heterogeneity in this time scale parameter. And it occurs such that regions at the periphery of this hierarchy, they tend to have faster decay, and regions at the top of the hierarchy tend to have slower decay. So this is a very influential paper from Murray et al. And this is also observed in fMRI experiments. So this is just a very recent example from human brain. So this is just published in bioRxiv recently, in which you also see core regions being more, in this case, well, it's more, it has a longer intrinsic time scales, and peripheral regions have shorter intrinsic time scales. So this was done in human, and it has been done before in the mouse as well. So across different modality and across different species, one observes this hierarchy of time scales. So it seems to be a very strong organizational principle of the brain. So I'm going to now jump into a different question. So what's the implications of this to brain dynamics? So one can look into this, for instance, using brain stimulation. So what happens if we stimulate regions that are at different levels of the hierarchy? If we stimulate regions that are at the top of the hierarchy and at the bottom of the hierarchy? So intuitively, one can think as hub regions that have many connections, that are strong connections. So if we have this linear intuition, we'd think that stimulation at hub regions would give rise to, well, maybe stronger and more widespread effects, whereas stimulation at peripheral region would be more local and weaker. So this was somewhat of an expectation if you have this linear idea as an expectation of these experiments. But it's not exactly what happens. But well, we had to test this first. So we did this experiment in fMRI. So this was led by Luca Cocchi. So 21 participants went to fMRI before and after TMS. And the TMS was a TBS protocol, an inhibitory TBS protocol, continuous TBS. And after stimulating, after inhibiting a peripheral region, what we observe is that this region becomes more functionally connected with other brain regions. But in contrast, if we stimulate a core region, it's FEF in the visual hierarchy, we observe that this region becomes less functionally connected with other brain regions. So yeah, our expectations were not good ones. So what we observed was actually these opposing effects in which you have one decrease and the other increase the functional activity. And we'd like to understand this better. And in order to understand this, I would like to propose a very simple model. So this model, it's similar to the previous one, but has an additional ingredient. Now we already assume that, so we start with the connectome, so now we use the human connectome to compare with the data. And now we assume that there is heterogeneity, that regions are not identical, but some regions are going to have faster intrinsic timescales, and hub regions are going to have slower intrinsic timescales. So this is already incorporated in the model. And in addition, we have this very, very simple network dynamics that incorporates the connectome and this heterogeneity in the natural frequency of the model. And finally, we assume that the inhibition is going to slow down the intrinsic dynamics of the target region. So everything else remains the same except for the intrinsic dynamics of the target region that becomes slower. And then we look at the effects of this. So if we compare the functional connectivity before and after this simulated inhibition, what we observe that after inhibiting a peripheral region, we have this increase in functional connectivity of this region with other brain regions. And then when we stimulate or we inhibit a core region, we see a decrease in functional connectivity between this region and the other brain regions. So this is what we wanted to reproduce. And this model also helps us to understand what could be happening. So one explanation is that we have this hierarchy of timescales. We have core regions that are hub regions that are already slower than other brain regions. If we inhibit this region, we slow down even further. This makes this region harder to interact with other brain regions and tend to decrease the functional connectivity between this region and other brain regions. And then in contrast, if we inhibit a peripheral region that's faster than other brain regions, this brings together these dynamics with other brain regions and tend to increase the functional connectivity, the interactions in the functional connectivity of this region with other brain regions. So those were two regions, V1 and FEF, that we had the data, but using this framework, this computational modeling framework, we can simulate the results of stimulating all other brain regions and try to understand what happens. And that's what we do. We extend this to 500 regions. And what we observe is that after we stimulate peripheral regions, there is a little bit of variability here, but in general there is a trend, a consistent trend that after stimulating peripheral regions we tend to see increase in functional connectivity, and after stimulating core regions we see decrease in functional connectivity following inhibition. So if we, instead of inhibit, we excite, and we assume that the excitation is going to speed up the intrinsic frequency of the target region, we actually see the opposite effect. So we see excitation of peripheral regions decrease the functional connectivity, and excitation of core regions increase the functional connectivity of the target region with other brain regions. So yeah, this is the opposite effect. So that was what I would like to cover today. So in conclusion, this hierarchy of timescales seems to be a very general phenomenon. It emerges just from the heterogeneity of networks. So even assuming everyone's identical, we can see that there is this hierarchy of timescales that emerge. And this is also consistent with how we understand the brain and how it interacts with the external world and with the internal body. We observe this opposite network effect. So this highlights the nonlinearity part of the brain, that linear intuitions is not always good. And we propose this very simple model that can reproduce and explain these opposite effects of brain stimulation at different levels of the hierarchy. And we like to propose this tuning curve that's more a prediction and has to be tested after stimulating other brain regions and see what would happen in that case. I'd like to thank my collaborators, Luca Koch, James Roberts, Michael Breakspear, and Andrew Zaleski. And I'd like to thank you for your attention.